Hello and welcome to our session titled Current and Future State of CAR T-Cell Manufacturing, Closing, Automating, and Preparing for the T-Cell Therapy Revolutions. I'm your moderator for today. My name is Xavier de Montlérat-Dujeu. I'm the Director of Innovations and Collaboration for Cell and Gene Therapy at Thermo Fishers Scientific. At Thermo Fishers Scientific, we're committed to make the world healthier, cleaner, and safer. And in particular, for cell therapy, we want to become your partner of choice to develop the next cell and gene therapy. And we want to do this, to do this, we have three main focus. One is really develop the new innovations of tomorrow, the best-in-class products that give you scalable solutions that will allow our customer to take their research to the clinic and make the manufacturing of this therapy more robust, more reproducible, and cheaper. The second thing we're focusing on is really around supply chain. These are complicated process that requires not only high-quality GMP manufacturing and regulatory support, but also guarantee of supplies. So we believe this is critical, and over the past few years, we've made significant investments in this area. And finally, and probably the most exciting, we are collaborations. We are doing a lot of collaborations. Our goal is really to develop products for researcher, drug developer. And so we strongly believe that working together in partnership through collaborations with our customer is the key to our success and making sure that we develop the right products, and as well as driving more robustness in the process that is critical to our missions. So we have active programs dedicated to this, providing early access to our technology, technical support, labs with our technology in those labs, and even co-develop processes. So please reach out, and we would love to work with you more and help you develop your process. Okay, now it's time for our speakers. So today we have four great speakers, and I'm really excited about. We have first speaker will be David Shaw, the principal scientist and group leaders, cell therapy process development at Genentech Roche. We'll then have Sean Cheng, product manager of cell and gene therapy at Thermo Fishers Scientific and developing automations and digital solutions for cell therapy manufacturing. Ben Schilling, who's a process engineer at Caribou Bioscience. And then finally, Evan Snyder, a staff scientist at Thermo Fisher, working on new expansion solutions for cell therapy. So what we will do is we will take a few questions after each speaker. And if there are more questions after, we'll take at the end of the speaks. So with that said, I'd like to introduce you David Shaw, principal scientist at Genentech Roche. David, the floor is yours.
Great, thank you for the introduction. Again, the title of my overview is Challenges of Autologous Neo-Antigen T-Cell Receptor T-Cell Therapy Manufacturing. I am a principal scientist and the head of cell therapy process development in the newly formed Cell Therapy Development and Engineering group at Genentech. The agenda for my talk today is a brief description of who we are and what we have accomplished, as I expect that Genentech does not present at this meeting often, or if ever. I will transition to what we are hoping to accomplish in the cell therapy field, followed by my general opinions about the challenges and mitigations for autologous T-cell therapy manufacturing, and then a summary. Genentech became a member of the Roche Group in 2009, and we claim to be the largest biotechnology company in the world. Genentech was founded in 1976 in South San Francisco, California, where the local bylaws allowed for recombinant DNA research, and we are still headquartered in South San Francisco. We employ approximately 13,500 employees in the US, and the South San Francisco site is the only Roche research site in North America. Roche was founded in 1896 and is headquartered in Basel, Switzerland. Currently, Roche has approximately 100,000 employees in over 100 countries all over the world. Roche has invested approximately $12 billion in R&D, which we claim is more than any other healthcare company. And we are currently focused on oncology, immunology, infectious diseases, cardiometabolism, ophthalmology, neuroscience, and respiratory diseases. We have 37 FDA breakthrough therapy designations. We have 32 medicines in the WHO model list of essential medicines, from 40 plus FDA approved medicines for serious and life-threatening diseases. We currently have over 90 investigational medicines in development, with greater than 1,150 clinical trials worldwide, and 570 of them are in the United States. We have 4 decades of transformative medicine, starting with protropin, or human growth factor, which was the first marketed recombinant protein. We then had a consistent commercialization of medicines throughout the years, including Rituxan, Herceptin, Avastin, Actemra, Tecentriq, Hemlibra, and Ocrevus, and those are the ones that I can remember. We hope to continue to bring meaningful impact to patients. However, even with all this posturing, this audience may have noticed that we do not have a cell or gene therapy in this timeline. This is what I hope to change and what I would like to introduce to you today. Genentech has partnered with Adaptive Biotechnologies to develop neo-antigen T-cell receptor T-cell therapy. In this business arrangement, Adaptive Biotechnologies is responsible for T-cell receptor identification and sequencing, while Genentech is responsible for T-cell receptor engineering, manufacturing, clinical trials, and regulatory filings. We are first exploring an autologous therapy, as the implementation is seemingly more straightforward, though the chain of custody and chain of identity is a logistical challenge. We are then dividing this autologous therapy into shared antigen and personal antigen programs. On the top of this flow diagram, we have a shared tumor-associated or neo-antigen, which is found in many patients. The goal is to build a library of shared or tumor-associated antigen T-cell receptors. The patients are then screened for those that express the neo-antigen or tumor-associated antigen, and the appropriate T-cell receptor is used to edit their T-cells. In the personalized neo-antigen T-cell receptor approach, the patient's tumor is sequenced, and the patient is HLA typed. A neo-antigen T-cell receptor is found in either the patient or a healthy donor, which binds the neoantigen expressed on the patient's tumor. The T-cell receptor donor is manufactured and used to edit the patient's T-cells. The T-cell manufacturing workflow is essentially the same for both the shared and personalized T-cell therapies, where we start with an apheresis of a patient, isolate and activate the patient's T-cells, edit the T-cell with non-viral gene editing, expand the T-cells, formulate and cryopreserve, and then infuse back into the patient. The main difference in manufacturing with the two approaches is that we can manufacture the T-cell receptor donor in advance with the shared neoantigen approach versus the T-cell receptor donors manufactured only after sequencing of the patient, which is in line with the T-cell manufacture with the personalized neoantigen therapy. As this overview is to highlight challenges, I'd like to share some personal opinions on what I've observed developing an early phase clinical process. From a process development perspective, since we are starting from autologous starting material, there is dramatic donor-to-donor variability. So it is sometimes challenging to determine if the process changes are due to donor variability or actual process changes. Current isolation and T-cell activation processes can be effective, but they are expensive and not always efficient. We decided to build our program with non-viral gene editing, as we know we could not have enabled personalized therapy using a viral vector. High efficiency, non-viral gene editing was challenging to implement. The graph on the right depicts our early data for percent knock-in of a neoantigen T-cell receptor versus T-cell expansion. The y-axis is 0% to 90% knock-in, and the x-axis is 0-fold to 75-fold expansion. We were able to find conditions to enable very high knock-in of almost 90% with reasonable expansion, as well as other conditions in which we had greater than 50% knock-in with much greater cell expansion. That leads me to finding effective gene editing conditions which allow for good cell health after gene editing, that could confer robust expansion with controlled phenotype. We have a lot of ongoing promising research to improve knock-in efficiency and ensure consistent performance across all our donors. While I'm not prepared to discuss that data today, we hope to be able to present that to you in the near future or perhaps at next year's conference. Autologous manufacturing challenges include many aspects of supply chain. Apheresis products are quite varied, and clinical sites are challenged to support many different sponsors, each of which has slightly different protocols. GMP reagents in the cell and gene therapy space are becoming easier to find, but surprisingly, the quality analytics and regulatory support sometimes requires a lot of work. Non-viral gene therapy reagents are not commonplace quite yet, and they are very, very expensive. As previously mentioned, the vein-to-vein logistics of autologous therapy is very challenging, especially for a company such as ours, who is more prone to or more familiar with biologics. Also mentioned previously, donor-to-donor variability makes process development, not to mention later stage process characterization and process validation, much, much more challenging than it needs to be. Later in this session, you'll hear Ben speak about ways to eliminate donor-to-donor variability, which I feel will be very impactful for the future of cell therapy. There is a lack of understanding and control over the manufacturing of autologous T-cell therapy, and we are working very hard to gain that understanding and control over our process. And finally, I'd like to just speak about competitive vendors. Currently, cell therapy is very expensive and unlikely to be first or second line therapy at these costs. There are a lot of companies vying to support the cell and gene therapy space, and hopefully some competition will help drive the costs down. So in summary, Genentech is partnering with Adaptive Biotechnologies to develop neo-antigen T-cell receptor T-cell therapies for both shared and personalized neoantigens. Our proof of concept will be demonstrated with shared neoantigens or tumor-associated antigens, but our goal is to bring personalized neoantigen T-cell therapy to patients so that more patients can be treated. There are many, many challenges with autologous T-cell therapy. We continue to work with cell therapy vendors around sourcing challenges, and COVID-19 has impacted the world as well as research. We agree that some challenges will be mitigated with allogeneic starting material, but our goal of personalized therapies will still have significant logistical challenges. And finally, I think a stronger understanding of T-cell therapy manufacturing will help mitigate much of these challenges. So thank you for your time. I'd be happy to take any questions.
Yeah, thank you, David. This was great. Few questions came about your process. Is it a modular process or automated? And what do you think of the difference between modular and automated systems?
So currently our process is modular. And my opinion around a fully automated system, while it may seem very attractive to have an all-in-one system that could potentially run your entire process, should your process actually fit into that system, that would be very attractive. However, I do feel that with all-in-one systems, you tend to, or you can be limited to your what can be carried out with your process, and that you end up trying to fit your process into that system, and you may or may not come up with the best process in the end. So currently, I'm a fan of modular processes so that we can insert the newest and best technologies to ensure that manufacturing is efficient.
Great, thank you. Your personalized approach is very interesting, but it seems to be quite a bit of a challenge to do. You know, you have to sequence the patients, get personalized donor DNA. What are the major hurdles to develop this and commercialize this type of approach?
Right. So I think that from a personalized perspective, you're right. The chain of custody, chain of identity, I think that's very similar to what is needed for autologous therapy. There's just yet even a further logistical challenge to track the actual manufacture of the donor DNA, since that can only go to one patient. So that is in addition to just the challenges around autologous therapy. So I do feel that those systems are either in place or being optimized by several companies as well as other vendors. So we hope to leverage that type of work. I think the major challenge for us for manufacturing the personalized therapies is just to ensure that the turnaround time can be adequate to support these patients. As we know, these are late stage patients. They've likely relapsed twice from their therapies already. And the time for manufacturing is extended if you have to sequence the patient, manufacture the donor DNA, and then manufacture the T-cell therapy. So I think that trying to do this as fast as we can while maintaining the quality that we need is definitely the big hurdle for us.
Yeah. Another one, is the non-viral gene editing mediated via electroporations in your process?
Yes, currently it is a electroporation process. I think that seems to be the most effective methodology right now, and for non-viral gene editing, in my opinion. Perhaps there will be newer technologies coming down the pipeline shortly.
Okay. Well, thank you, David. So if you have more questions, we can address those questions at the end of the sessions. Now we're going to move on to our next speaker, Sean Cheng, Product Manager of Cell and Gene Therapy at Thermo Fisher, and he'll tell us about automation and digital solution for cell manufacturing. Sean, the floor is yours.
All right. Thank you, Xavier. Hello, everyone. I'm Sean Chen, and today I'm going to introduce Thermo Fisher solutions to support closed, modular, and automated cell therapy manufacturing. And as many of you know, cell therapy has been rapidly evolving in the past 5 to 10 years. But as David and Xavier mentioned earlier, there's challenges in every aspect of the industry today. From supply chain, logistics, technology, process, all the way to regulation. And Thermo Fisher has been dedicating to help customers overcome the hurdles from every angle of their productions. But obviously, for a lot of our customers, the top 3 challenges are still manufacturing, manufacturing, and manufacturing. So let's take a look at what's the main issues in cell therapy manufacturing today. As David gave a really good example of autologous T-cell therapy, as you can see on the left, a general T-cell therapy workflow includes T-cell isolation, activation, gene modification, all the way to harvest and cryopreservation. As we listed on the right-hand side, it is a very complex and labor-intensive process, which requires a lot of open manipulation. And it is also very difficult to synchronize and harmonize different instruments, products, and to make the whole workflow traceable and compliant to regulatory requirements. So based on these issues, we understand that customers are looking for 3 main solutions. First, the automation system to reduce labor and human error. Second, a closed system to minimize contamination. And third, a modular system to maintain flexibility. And this is exactly what Thermo Fisher has been providing. We are building best-in-class, closed, modular, and automated instruments in combination of the reagents, protocols, and also analytical tools. While we can physically integrate them, we are also enabling digital connectivity of the workflow to support full automation and data integrity. And as Xavier mentioned earlier, now we have a whole PD team in our new cell and gene therapy business unit to provide more collaboration opportunity. And the whole PD team is optimizing and integrating all the products in the entire workflow and provide the clinical scale protocols to our customers. Now I would like to provide a little bit more details on the digital side. On the left-hand side, you can see we believe the end goal of a mature manufacturing is to manage and integrate the production layer through a DCS controller system to ensure traceable and secure data connectivity. And to support this architecture on the right-hand side, Thermo Fisher is developing an off-the-shelf software library to connect our instrument directly to Delta V DCS system, which is one of the most widely used DCS system in the field. And by doing so, we're building a strong foundation of the production and control layer for cell therapy and support the integration of workflow and data. And now, after introduction of our overall vision, I would like to go deeper in our solutions for the whole vein-to-vein cell therapy manufacturing, especially focus on the first half of the process, which is day 0 to gene modification, and address customers' pain point on cell isolation, electroporation, and gene modification. And I would like to introduce our first solution, the rotia system, which is a closed automated cell processing system specifically designed for cell therapy. The counterflow centrifuge technology provides a process flexibility and allows customer to perform cell isolation, wash, concentrate, and buffer exchange. And the unique chamber design allows a very low output volume, in the meantime, provide very high recovery and viability. And we have implemented the OPC-UA interface in rotia to support digital connectivity. Here's another overview of the whole system. On the left-hand side, we have a compact, a small footprint instrument, which can be easily integrated into your current process. In the middle, we have closed single-use kit, which has flexible input and output ports, enabling the fast and multi-purpose process. On the right, we have a user-programmable software interface, which allows you to create your own protocols. In the meantime, Thermo Fisher provides preset protocols for T-cell processing. We also have a live camera, which can support a real-time view for your cells being processed in the chamber. And for day 0 process, rotia is capable of supporting PBMC isolation and T-cell isolation. And this is a summary of PBMC isolation using a preset protocol with ACK buffer. So on the left, you can see a very robust RBC depletion and PBMC enrichment after rotia process. On the right, the PBMC isolated by rotia have similar fold expansion and phenotypes as compared to the cells from traditional phyco separation. This indicates the cell quality after rotia is as good as the manual process. Also for day 0 isolation, our PD team has been working on using rotia to do T-cell isolation in combination with our Dynabeads technology. As you can see, the setup and the process on the upper side. First, we use rotia to wash frozen leukopak to remove DMSO, platelets, and serum. And then we use rotia to combine our Dynabeads, CD3 and CD28 Dynabeads with the cells. And after incubation, rotia collects the target cells bound with beads and elucidate non-target cells. As you can see from the lower panel, we can efficiently enrich CD3 positive T cells while removal of the other cell types, including B cells. We're still optimizing the protocol and for more a pure T cell purity after the isolation. And I would also like to emphasize this application, the intention of this application is not to replace our magnet system, but to offer an option for the customer who wants to use rotia to simplify their process. And after cell isolation, the next challenge in the workflow would be a pre-electroporation process. So here's the example of using rotia to wash exchange buffer and concentrate T cells in electroporation buffer. As you can see from the bar graph, we can achieve very high viability and recovery while concentrating the T cells in low output volume. And the output volume used here in this study was around 10 mil. But we also have data showing rotia can go as low as 5 mil for the output without compromising viability and recovery. And the other layer of pre-EP process is the payload addition. So here's an example of the closed connection that our PD team is working on between rotia and our new electroporation system, which I will introduce in the next slide. As you can see in the middle, the concentrated T cells from rotia can be collected and mixed with the payload in a closed tube. And then the cells and the payload will be transferred to our EP system, winning a closed and automated manner. And I would like to emphasize our PD team is capable of providing the support like this kind of consumable to specific customer needs through different collaboration opportunities. Now I would like to go deeper on our electroporation system for supporting the gene modification from R&D to manufacturing scale. On the left, we have our neon electroporation system, which is a smaller volume system to enable your easier optimization. And the unique tip design provides a very robust and efficient gene editing efficiency, including multiple knockout and knock-in just one zap on T cells. In addition, as you can also see from the dot plot, the knock-in efficiency can be drastically increased with the help of our gene editing buffer. Now we are seamlessly transfer the capability of Neon to our Xenon system, which is a new large volume electroporation system and will be launched soon. And this is a closed automated system which can accommodate 1 ml single shot or 5 to 25 ml flow through reaction. The gene editing efficiency of this system is similar or even better to the Neon system. All the consumables and reagents, including gene editing buffer, will be GMP grade. And we will also provide a digital connectivity through the OPC-UA interface directly to Delta V system. Lastly, similar to a lot of our instrument, Xenon allows the customer to change parameters, for example, voltage and pulse number and pulse width for a much easier optimization. And these are a couple examples of using Neon and Xenon to perform gene knock-in in T cells. As you can see on the right, the large volume Xenon system can achieve a similar or better knock-in results at multiple different loci as compared to a smaller scale Neon system. In addition to rotia and Xenon system, Thermo Fisher has instrument solutions in every step of the workflow. For example, we have large-scale Dynamac magnetic system for cell selection and bead removal. The next generation automation version of the Dynamac is under development. For cell expansion, we have HeroCell incubator specifically designed and certified for clean room use. We also have high performa rocker and stir tank bioreactor for dynamic expansion. And Evan later will provide more details in our expansion solution in his presentation. And for cryopreservation, we have CryoMat control rate freezer with very robust and consistent temperature control. And again, for all the instruments in this workflow, we will provide software solution to connect them directly to Delta V system. And finally, I would like to re-emphasize Thermo Fisher's vision and capability on cell therapy manufacturing. By building the closed modular systems and combination with superior reagents, analytical tools, and software solutions, we promise a scalable and flexible workflow with traceable and repeatable performance. With that, I will thank you for your attention, and I will take any questions.
Great, thank you, Sean. So actually, there's a few questions come in. So regarding your approach using Delta V in the modular system, so have you demonstrated the complete manufacturing workflow through Delta V, demonstrated the automations. And have you done this internally, or have you done this with collaborators?
Yeah, we actually have done this internally. We have internal experience of using Delta V to control rotia, our magnet system, and our rocker system to perform a lentiviral CAR T workflow from end to end. And this was our early effort for a proof of principle study. And the software codes and the interface used in that study was early phase prototype. But now we're using this experience to build our software solution product to our customer, like I mentioned earlier.
Great. You mentioned few applications with rotia, but what other applications you've seen people using rotia for?
Yeah, rotia is a very versatile instrument. The technology is based on, like I mentioned, the counterflow centrifuge. So it's based on different cell size, different cell density. So it can perform a lot of different applications in addition to the ones I introduced, like cell isolation, wash and concentrate. We have some internal or external data that maybe we can perform monocyte separation from, separate them from the T cells, which is still being optimized, but the results pretty good right now. And we have some stem cells data from MSC workflow that maybe we can separate some aggregates from the culture to the singles, separate them from the single cells. So there's a lot of opportunities of the rotia instrument, and you can find some of the introduction or some of the application rotia can perform on our website.
Great. Another one is, can the rotia kit be used to formulate the final cell products with cryomedia containing DMSO? Are the rotia kits DMSO resistant?
The short answer is yes. We have extractable, leachable data and testing on DMSO, and we have applications specifically designed for cryopreservation or buffer exchange to cryopreservation. So yes, rotia can support your final step of harvest and buffer exchange to your cryomedia, and yes, rotia is resistant. The kit is resistant to DMSO.
Yeah, and if I may add, I mean, we've done also extractable and leachable testing with DMSO, and it passed the regulatory guidelines. It's including into our regulatory support file, for these applications. So great, thanks, Sean. Another question, might be difficult to answer, but how does the gene editing buffer increase the knockout and knock-in efficiency? And is this buffer commercially available?
Yes, this buffer is commercially available through a Xenon system. So it's a Xenon consumable reagent. And I'm not sure how much I can tell about the background of how this reagent can increase knock-in efficiency, but it's a lot of the testing and formulation from the R&D side, and specifically targeting for the knock-in efficiency of the homologous recombination. So that's the key help of the knock-in efficiency.
Great, thanks, Sean. Just a little correction, it will be commercially available as with the Xeon launching later this year and early next year, but right now it's available through collaborations. Any other questions I can see here? Oh, what is the highest cell concentrations rotia can achieve in the output?
Yeah, we have good data from R&D and also external customers that the highest cell concentration is 400 million per mil that rotia can achieve to the output.
Great. So another one. So I'm going to read it. So I've tested the rotia and got great results for MSC concentrations and volume reductions, but the connection to the rotia is still very limited and require welding of PVC. Is this something being worked on?
Yes. Our PD team and also Rotia R&D team has working closely on connecting to different upstream and downstream application for Rotia. So we see some difficulty, especially on the MSC side, like you mentioned, we have to weld it on maybe a static culture vessel. So that's something we're working on. But yes, like I mentioned, Rotia has a flexible input and output ports, and with PBC weldable tubing, so it's easier to connect to a lot of different bags, different consumables from different vendors.
Yeah. And has rotia been used with adipose tissue for MSC isolation?
That's a good question. I'm not sure about that specific application. I have to go back to our R&D team and hopefully, I can answer it at some format.
If I imagine, and I think you mentioned before, right? Rotia is separating cells based on their density. So you could assume that any cells that are, that have a different density than the other cells in the environment should be able to be separated with rotia.
Yeah, correct.
And also we see growing applications, with rotias, and we're posting on our website. Okay. Well, thank you very much, Sean. We're gonna move on to our next speaker. Very pleased to introduce Ben Schilling, a process engineer at Caribou Bioscience. Ben, floor is yours.
Great. Thank you, Xavier. My name is Ben Schilling. I'm a process development engineer with Caribou, and I will be talking about our large-scale allogeneic CAR T-cell manufacturing. So first off, I'll just give you an overview of what I'll be talking about. I'll introduce Caribou Biosciences. I'll talk about why we're interested in allogeneic CAR T therapies. I'll go over our pipeline, our platform, and our process, and specifically talk about our next generation CRISPR technology. I'll wrap it up with some of the pain points and some of the focus areas, and then finally I'll summarize everything. So to introduce Caribou Biosciences, Caribou was founded in 2011 by Jennifer Doudna and Martin Yannick. Our intellectual property includes our CRISPR Chardonnay technology, which can be paired with either Cas9 or Cas12a proteins. Our current aim is to leverage this next generation genome editing to develop cell therapies for oncology. Besides our pipeline, we also have a strategic collaboration with AbbVie to develop 2 other CAR T programs. And a big highlight from earlier this year was closing a Series C funding round. So I'm going to talk about allogeneic CAR Ts. We have been talking about autologous CAR Ts earlier in this presentation. There's obviously been very impressive clinical responses, albeit with some caveats that have already been discussed, but just to reiterate them, the long vein-to-vein times, the fact that bridging therapy is often required due to these vein-to-vein times. There is the possibility of manufacturing failure. There's definitely a variability in T cells from different donors, and of course the high production costs. So allogeneic therapy provides this dream, this ideal of making off-the-shelf products that would allow us to build up an inventory such that the product could be readily available. This would mean that bridging therapy wouldn't be required. We could also have more uniform T cells pooling from healthy donors, and a more efficient manufacturing process. We could manufacture multiple doses in one manufacturing campaign. This would also allow us to reach some of the patients who might be ineligible for autologous therapy, and it opens up the door for repeat dosing. So to go over our current pipeline, our program CB10 is an anti-CD19 and is currently in phase 1. The next 3 programs, we have a CB11 program, which is targeting BCMA, is going through the last developments before an IND enabling study. And then further on down the pipeline, we're developing a therapy for AML. And we're actually finding ways to edit other cell types besides T cells, our INK cells, which is an iPSC-derived NK cell. And we're hoping to target this towards a solid tumor indication. So let me walk you through our process. The CB10 allogeneic CAR T manufacturing process uses 4 different critical raw materials. The leukapheresis from a healthy donor, as I alluded to earlier. We also use an adeno-associated virus for the site-specific gene insertion. This program, the CB10 program, uses the Cas9 protein along with chRDNA guides. So the gene modifications, we knock out the TRAC locus and knock in an anti-CD19 CAR, and we've also knocked out PDCD1, which encodes PD1 to decrease T-cell exhaustion. These edited T-cells are then expanded. After the expansion, we perform a TCR alpha-beta depletion to remove any residual unedited TCR alpha-beta positive cells. Then it goes into the final formulation and cryopreservation. The next program in our pipeline is CB11. This is also an allogeneic CAR T, and in this case, our target is BCMA. So we knock in an anti-BCMA CAR into the TRAC locus, and our second edit that we perform is to knock out class 1 and knock in a beta-2M HLA-E fusion. The benefit of doing this is to prevent or at least reduce T-cell clearance and NK-mediated clearance of CAR T with the goal of encouraging more persistence. And this program will use Cas12a protein as opposed to Cas9, also with our chRDNA reagent, and I will discuss those in the next slide. So our CRISPR technology is using these hybrid RNA-DNA molecules, so the chRDNAs. And so this is based on the RNA guide, but some of the nucleotides, the RNA nucleotides, are replaced with DNA nucleotides. And we have found that this can dramatically improve the specificity. So it will allow us to maintain our on-target editing, but dramatically decrease our off-target editing. And this system can be applied for T cells, NK cells, and iPSC derived versions of both of those. This is actually, I think, what separates Caribou. We have a lot of intellectual property in this space, and we've put a lot of our effort towards getting this really highly efficient editing system. So just kind of double down on this with our Cas12a protein combined with the chRDNAs, we're able to, as I mentioned, achieve high efficient site-specific insertions. We're reaching insertion efficiencies of 50 to 80% of the gene insertion. And as far as the gene knockout, can be anywhere 90% above, 90% and above. And this is kind of the core of our technology that drives our cell therapy programs. So to dive a little bit deeper into this comparison between the RNA guides and the chRDNA guides, here's a demonstration of Cas9 and Cas12a systems where we've identified a few target genes, and we're comparing the RNA to the chRDNA, and we're looking at the on-target editing percentage as well as the most common off-targets. So if there's the 3 most common off-target percentages. And so for the RNA guides, you can see that we obtain a very high percentage of gene editing. For AAVS1, for instance, it's above 90%. However, we also observe a large percentage of off-target editing using this guide. However, when we optimized our guides, mixing in some of these DNA nucleotides, we're able to dramatically reduce our off-target editing and still maintain the high editing percentage of the desired targets. So now that I've talked about the editing, I want to go over how this impacts our cell growth and our expansion. So here I'm showing you a large scale. This is a representative run from our CB11 program, which receives 2 edits and 2 gene knock-ins. And I'm showing you the population doubling and then over the days of production, the origin is starting from day 0. I can't tell you how many days our process is, nor can I tell you how many population doublings. But just to give you an idea, we're comparing the edited cells to the wild type or mock T cells. And you can see that the red line there is a lag that from the editing and from the viral gene insertion that slows down the growth at the beginning of the process. But by the end of our process, our CB11 cells have caught up to our wild type cells. So now that I've gone over the editing and our cell growth, I want to talk a little bit about the pain points and the focus areas. Of course, as we mentioned in this symposium, the complex logistics and the supply chain required for T-cell manufacturing definitely present a unique challenge. The sourcing of the raw materials, the GMP leukopaks, Cas proteins, virus, the Chardonnay guides, each of those items has a long lead time. They can have manufacturing challenges of their own. They can have manufacturing failures. There's definitely quality issues from one vendor versus another that we've observed. So it's very critical to find the sources that we trust for each of those. Another challenge is obtaining large volumes of media and supplements, given that we're trying to grow an allogeneic CAR T. We're trying to make several doses in each manufacturing run. We're talking in the scale of billions of cells, so that requires large volumes of media, which means you have to be prepared to have the media on hand and establishing the supply chain for that. And then, of course, as you all know, the global shortage of medical-grade plastics due to the pandemic and also due to just the increased activity in this field. So then I will talk about some of the focus areas. One of them is the recapitulation of the benchtop scale results in larger real-world systems, basically performing editing in a small-scale setup and finding the best Chardonnay guides, but then taking that into a large-scale manufacturing system where you're no longer editing just millions of cells, you're looking at billions of cells, and you want it to be a closed system, and you want to maintain the viability and the expansion of the cells after they're edited. And so there's a balancing of multiple variables. And then I want to bring up that we have to harness analytics to make these process decisions. And as your scale increases, we've learned that all of the independent variables influence one another. The way that you activate cells can influence how well they're edited or how they're transduced, and not to mention editing and transduction will affect the cell's expansion. And then, of course, the expansion of the cells can guide them from differentiate them from one phenotype to another. And so it definitely takes a DOE approach to incorporate all of these variables together. So in summary, allogeneic CAR T manufacturing hopes to build on the promising results of autologous CAR T therapy. We believe that CRISPR genome editing technology will enable us to generate these allogeneic CAR Ts to remove the track locus and insert our CAR, but also to make multiple edits that can allow the proliferation of these cells, can also decrease exhaustion, and encourage their persistence. And so finally, I want to say that I think producing large-scale off-the-shelf cell therapy products derived from healthy donors is feasible. And I will take any questions.
Great. Thanks, Ben. And thanks for reminding your pain point. We understand them and trying to do our best with the supplies, at least, and logistic. So we have a few questions. So first of all, are you multiple gene edits are done in parallel? And if so, what's the translocation risk if you do this? And how do you mitigate that risk?
Yeah, this is a great question. So our current process is using a staggered editing approach, but a parallel editing approach is also in development. So there's I think benefits to each of those strategies. So the parallel would be to enable us to incorporate more edits in our product, and then there is a growth disadvantage during the editing process. Editing cells will cause, during the buffer exchange, you'll lose cells, and so you kind of want to minimize the number of edits that you do throughout your process. So far as like the translocation risk, I'll address how we have approached this problem. So we've created a couple of different assays to identify where translocations are happening and quantify how often they're happening. And so to mitigate the risk is really to evaluate the specificity of the Chardonnay guides, as I showed earlier. So creating the really highly efficient guides that maximize your on-target editing, minimize off-target editing is one of the ways that we mitigate the risk, and then we want to quantify it. So we go and look at which genes might be being translocated, and then identify are these oncogenes. We're also looking at not just translocation, we're also looking at deletions, and then inversions in our Cas9 system. So just looking at what are all the things that could possibly happen, and then figuring out what is the risk with each of those, and finding ways to minimize them.
Great. And we always talk about off-target rates, and the Cas12 seems to have better result and profile than Cas9. But what is an acceptable off-target rate?
Yeah, that's another great question. I think we're still trying to figure that out, in that we're waiting. That I don't think the agency has provided like a specific percentage, like you have to be below this percentage. Yeah. The idea, I think, is to try and generate as few off-target events as possible. With the genes that we've been targeting so far, we're getting with Cas12a system below 0.1% off target, and so we think that is acceptable. But I don't know what that number will be when it's all said and done. Yeah,
that was a tough question, a tough one to answer. So the graph showing the lag and then expansion is interesting. Do you have any hypothesis of why the lag is longer and then kickoff? Have you looked at that in more detail?
Yeah. So we think that the lag can be due to two reasons. The electroporation of the cells does produce a lot of stress in a short term for our cell expansion. And then also we're using a viral transduction method. So adding virus to our culture, at a high enough concentration to get the gene transduction. We have seen a dose response to adding more virus will inhibit growth. We also are hypothesizing that knocking in the CAR receptor can give kind of a tonic signal, a growth signal that might explain why it catches up towards the end of our process.
I see. Okay, so the lag was compared to a wild type that was not electroporated, that was not treated. Oh, okay. Oh,
yeah, that's
okay. Great. Well, thank you very much, Ben. And now we're going to move on to our next speaker, Evan Znajda, I'm trying to find it. Evan is a staff scientist at Thermo Fishers, and is actually based in Buffalo, New York right now, and working on developing new expansion solutions for cell therapy. Evan?
Yeah, thank you, Xavier, and thank you all for attending. So today I'm going to use my time to discuss current challenges in cell therapy that can be addressed by allogeneic workflows, and introduce some strategies to potentially increase therapeutic success. So I just want to start with a little bit of background on cell therapy based on clinical trial activity. So over on the left, you see a pie chart displaying the clinical trials by cell type, in cell therapy, that is. And you can see that T cells are still the biggest chunk of this at around 40%, and NK cells, which I have some personal interest in, are around 12%. But this 12%, even though it's low here, does represent a lot of growth recently. And then moving to the center, you can see another pie chart, but here it's looking at company profiles based on autologous and allogeneic interest. And you can see that 2/3 of the companies focus solely on autologous clinical trials at the moment, while 1/3 has at least some interest in allogeneic therapies, which represents a lot of recent growth. And then finally, on the right, we have the distribution of clinical phases based on cell type. And the dark blue and light blue bars are early phase trials, and you can see these dominate for all of the cell types. And it's not very surprising given that this field is booming and there is not a lot of success to get to phase 3 and beyond at the moment due to a whole slew of challenges, some of which I've listed here, starting with the time between diagnosis and the actual first dose administration, which, right now it's a pretty big ask of a lot of the patients. And then moving on to safety and consistency, we obviously want to prevent contamination and adverse effects. And then a big one, which has already been alluded to a couple times, is the cost to the patients. We need to significantly lower this in order to be able to reach a larger patient population. And then the final two can kind of be grouped together under like obstacles to efficacy. And donor material is kind of a crapshoot. We never know what we're going to get. Oftentimes, these cells don't respond very well. I'll get into that a little bit more in a couple slides. And solid tumors pose a lot of obstacles to these cell therapies. But many strategies have been developed. I've listed several of them here. And this ranges from, things kind of tinkering with the manufacturing process all the way to fine-tuning the gene editing strategy, kind of like what Sean and Ben talked about respectively. And if you look closely at these solutions, I kind of find a few major buckets that I've listed here. First off, closing and automating the workflow. This could improve safety and consistency and reduce the labor requirement, thereby reducing the costs. Second, applying allogeneic workflows, which again, we've already talked about a bunch in this session, but the way this could help is that we can produce these therapies at scale and they can be off the shelf, reducing both the time to treatment and potentially the cost. Not to mention that if we control the cellular input and hand-pick high-quality cells to input into these manufacturing processes, we should see an improvement in the cellular output from performance and maybe an efficacy standpoint. And then the final one listed here is a deeper understanding of the biology. And this is both on the side of the cell therapies themselves and also on the side of the target cells and the microenvironment. We know that there's responders and non-responders. If we can start to understand what differentiates, what signaling or pathways that are turned on differentially in responders, perhaps we can use this to convert non-responders into responders. So along those lines, like I said, we already know that patients with chronic disease often have less effective cells when they're turned into cell therapies. And a lot of this can be explained by the differentiation pathway of T cells. So T cells start off naive, and then as they become activated and continuously stimulated, they move through early memory to effector and eventually become exhausted with constant stimulation. And we want to avoid the effector and exhausted cells on the right side of the spectrum, as these aren't usually associated with very good clinical outcomes due to the fact that they don't show a lot of proliferative capacity or engraftment, and oftentimes they become senescent. Whereas those early memory cells towards the left, those do show a high self-renewal capacity, persistence, engraftment, and immunostimulatory cytokine production. So as a biotech, we need to develop workflow solutions to drive these T cells towards the left side and maintain early memory cells and delay differentiation into effector and exhausted cells. And one of the initial ways we thought, this made sense was through the culture medium. because it's in direct contact with the cells throughout expansion, and because it's pretty easy to manipulate in a way that could possibly turn on and turn off certain signaling in the cells. So our media of choice for T-cell therapy had generally been Optimizer. And this is because it is serum-free, which allows for, a little bit safer and more consistency, as well as a guaranteed supply chain. And it is also phenol red-free, which is automation-friendly. And maybe most notably, this media has already been used in the production of clinically approved cell therapies. So therefore, we know it can overcome many of the performance and regulatory challenges. So with that in mind, we tried to use this as a platform and manipulate the formulation a little bit in a way that would drive cells towards early memory and delay differentiation. So we call this media Optimizer Pro. And I'm going to just go into a couple of slides of data comparing Optimizer Pro to the parent Optimizer. And for the purposes of the next few slides, all this data was normalized, comparing, so we get a direct comparison of Optimizer Pro to the parent. But I should mention that all of the results, we did see robust results for all of the donors and the media. And this was done with 6 healthy donors and for 17 days in order to see how these cells behaved over a longer period of time. And you can see here, the dark black line represents the average of the 6 donors grown in the parent Optimizer media. The gray lines represent the individual donors grown in Optimizer Pro. And the light blue line is actually the average of these 6 donors grown in Optimizer Pro. So by day 10, we saw an average increase in proliferation of about 20%. And at this day, we also saw that all 6 of these donors did show some improvement in growth. And this effect actually becomes more pronounced over time, to where by day 17, we get about a 2-fold increase in growth. There was no difference in viability. Both the media and all of the donors were above 90%. There might have been a slight increase with Optimizer Pro, but it was nothing significant. So moving on to early memory marker expression. So here we looked at 2 different panels, which were pretty similar. One was CD62L and CCR7, and one was CD62L and CD27 positive. So the blue bars are actually the double positive staining cells, and this is normalized again. So any value above 0 represents an increase in the early memory population for cells grown in Optimizer Pro. So at day 5 and day 10, we see about a 10 to 20% increase in the Optimizer Pro populations of early memory cells. And then by day 17, we don't really see much of an increase, with basically the same population distribution. So we see the same amount of early memory cells. So if we put this together with the previous slide, it seems that early retention of these central memory and stem cell memory cells at day 5 leads to an increased proliferative capacity or self-renewal capacity. And by day 10, we actually have more cells, and a higher percentage of those cells are still early memory cells. And then this results almost in a compounding effect, which results in twice as many cells by day 17 total, and the same percentage of those cells is early memory. So therefore, we have twice as many of the desirable early memory cells out to a pretty far time point. Another hallmark I mentioned was the cytokine production. And here I am specifically showing IFN gamma release. And IFN gamma is an important catalyst in both the innate and adaptive immune responses. And again, this data is normalized. So all of these bars represent an increase in IFN gamma for each of the individual donors. And you can see the average increase was about 187% across these 6 donors. And this was done at the end of the expansion time. And even if we get rid of the donor 659, which seems to be a potential outlier, there's still a 55% increase in IFN gamma release for the other 5 donors. So this is really exciting to us, just given how important the cytokine is. So if we bring it all together, more cells with more immunogenic properties should result in a more robust targeted immune response. At least, that's the hypothesis. We have not verified that yet. This product just launched pretty recently. But if we're talking about going to the clinic, we also have to make sure that this product can perform at scale. All of the results I just showed you were developmental and feasibility work. So it was mostly done in G-Rexes and in flasks. but we also want to make sure it performed in rocking motion bioreactors and stirred tank bioreactors. So for this purpose, we looked into the Zuri and the stirred tank reactor, but I wanted to get into the actual benefits of these are listed here on the left. They're scalable, they allow for pretty intimate controls of the environment, including agitation and the gassing and liquid strategies, and they can be pretty easily assimilated into any workflow because they can be closed and automated and digitally integrated. So we started with the rocking motion bioreactor here, and this was done in a Zuri. And we activated the cells with dynabeads for 3 days in static culture and then inoculated on day 3 for the purposes of this experiment. And this was, again, a direct comparison between Optimizer Pro and Optimizer, and it was carried out for 17 days. We have repeated this with several donors, several different times, and we see these very similar results. These results are probably fall in the middle of the spectrum of results we've seen. We always, and I should also mention, this did involve perfusions since we were going out so far. We always tend to see a bit of a divergence around day 8 or 10 comparing the two media. We start to see the Optimizer Pro outperforming the Optimizer, and then this becomes more pronounced over time, similar to the small scale. And then looking over toward the right, this is also, again, associated with an early retention of those central memory and stem cell memory cells. And here we have the staining of CD27 and CD62L positive, and this is normalized data. So we see at day 5 and day 10 about a 6 or 8% increase in early memory cells. So it's the same kind of story as was in small scale. And then moving on to stirred tank reactors, this is a bit of a newer workflow for us. And I think I already tend to prefer these over the rocking motion bioreactors for some of the benefits I've listed here. They often have a smaller footprint with the same volumes being compared. There's no dead spots that you can find in the bag sometime in the corners and around the sensors, and you have even more intimate controls over the whole environment. You can make these cells extremely happy. And not listed here is that these are even more scalable than the bags usually are. So for this, though, we have done media comparisons and we have done perfusion, though most of that work is fairly preliminary and we aren't comfortable sharing it yet. Here, I'm just showing the performance of Optimizer Pro in 2 different vessels, the stirred tank reactor and a static benchmark, the G-Rex. And you can see, just looking at and this was done in fed batch mode, and you can see looking at the results here in fold expansion that there is a divergence at around day 6. And again, we've done this several times with several different donors, and this is very reproducible across donors, healthy donors too. And you can see that over time, this becomes more pronounced. There is better growth in the stirred tank. These tend to be longer lasting. And then obviously with perfusion, we can keep them going forever and very robust growth curves. And I'm looking at the early memory, or just the phenotype in general over here based on CD27 and CD62L staining on the right. Here, this is not normalized. This is simply looking at, in the dark blue bars, central memory phenotype, and in the light blue bars, an effector phenotype. And you can see that the dark blue central memory cells occupy very large chunks of the population, and the effector cells occupy a very small percentage, which is a desirable phenotype to us. So just to show that basically we're getting very good growth and we're maintaining the right phenotype, a desirable spectrum of these cells. So we're pretty excited to keep on working in these vessels and keep developing products towards allogeneic cell therapies. So with that, I just want to conclude with a couple of really important messages. And it's going to kind of reiterate some of the things that were said by the panelists as well as Xavier a couple times. We all know that the cells are the drug and the process influences the efficacy. So what has to be considered is everything. When you're creating a workflow, you have to really think about all of the devices and the reagents, the timing, every single nuance to every step in the workflow has to be carefully considered because it can all affect the outcome, which in turn affects the efficacy. So with that in mind, we're super ecstatic to keep on developing products for allogeneic therapies, which seem to have a ton of promise to save a lot of lives. And the best way for us to do that is to partner with our customers directly. If we partner during development, it kind of eliminates the guesswork and allows us to almost ensure that the product will work the way the customers want it to work in their hands, in their workflow. So with that in mind, if you have any gaps or any kind of questions around your workflow, please reach out. We love working very closely, and we flex to you. So with that, I'll kick it back to Xavier for some more discussion.
Great. Thank you, Evan. Love your steer bioreactor data. I think it's very exciting for the field. Okay, we have a few questions here. Has the Optimizer Pro been used to expand diseased donor cells?
Yes, during development, we did expand about maybe a dozen or 15 different donor samples, diseased donor samples, that is. And we did see an average increase in expansion of about 20 to 30% by day 10 compared to the parent medium. But the results were extremely inconsistent. And we saw maybe about just over 50% of them showed extremely favorable results, up to like 300, 400% increase in growth. But then we saw the other 45% actually were a little bit below the parent optimizer. So for now, this medium, while it can work very, very well for diseased donors, it's better suited in allogeneic therapies that use healthy donors at the moment, until we understand what determines how the cells will respond and what determines which cells will respond, which is work that we have ongoing.
Got it. With this new media, Optimizer Pro, is there a difference of perfusion schedule? For example, if you put on a Zuri platform, is there a difference between Optimizer Pro and Optimizer on the schedule?
For what kind of schedule? Oh, perfusion. Yeah. There's no difference. It's for both in both rockers and bioreactor and stirred tanks. We use a pretty standard perfusion schedule. Usually, starting off when the cells hit 2 million cells per ml, we might do about a 25% volume per day, and then we gradually carry it up there. And I think usually around maybe 15 million cells per ml, we might hit a little over a liter per day. But it's a pretty standard, and we use the same, at the moment, we use the same perfusion schedule for both Optimizer Pro and the parent media, though this may change in stirred tanks later. We're still optimizing that a little bit.
Great. Now, not necessarily related to this talk, but maybe to your experience in expansions, what do you think is likely the optimal culture size for scaling up iPSC culture? As you need to balance the passage number with efficiency of scale and the accessibility to media.
I haven't done anything with iPSCs yet, so that question, okay. Yeah, that's on the roadmap, but not for a little bit.
Yeah.
And then just with regard to T cells and NKs, T cells, people don't generally go over a liter. So if you're doing iPSC-driven T cells, you don't generally go too much higher. NKs are all over the map, so it might change depending on what you're trying to drive the iPSCs into.
I know it was a tough question. I should just add also, we have multiple groups working on different systems, and we have a group actually that just recently launched STEM Scale, which is our iPSC culture media for suspensions. As we preparing, and that's kind of this talk here, the next generations, and iPSC is definitely around the corner. Okay. Maybe, so I think we can open the floor to all the speakers now. Thank you, Evan. And we have 2 questions for the speakers. One is, and I don't know, I would say, let's say for Sean, what technology does Thermo have for process monitoring? Sean or Evan, actually.
So for right now, we are looking to different technologies for inline analytics. We understand that in-process monitoring or analytics is very important for monitoring the quality of your expansion. As for now, Thermo Fisher, we right now don't have an automation system to do so, but we're looking into the opportunities. But maybe Evan, you have more experience on the inline monitoring in your process?
Yeah, at the moment, our the The high performa controllers, the G3 and the GX would be where I would lean to, which can monitor all of your gassing, your liquids, basically that kind of thing. We are developing and we are looking into technologies for biomass and such. Every day we're trying out different things for biomass and different byproducts, but we don't currently have anything.
Great. Thank you. Now, a couple of questions for Ben, and I realized the IPSCS question could actually go to Ben as well, since they do this. So Ben, same questions around iPSC culture. What do you think is the optimal culture size for scaling up the iPSC culture?
Yeah, that's actually something that we're still developing. We're as we speak, we're trying a few different culture sizes, and we're trying to optimize the size of the culture, the ratio of feeders to our inks, and like creating workflow. And we're trying to, we have had some promising results in flasks, and we're trying to see if we can move to static bags and beyond, but still figuring out the way to balance all of the different variables involved.
Got it. And so are you scaling up the process right now, or are you still working in fairly small volume for your iPSC?
Yeah, so we're I think we did like the first feasibility test, so it's definitely an ongoing investigation.
Got it. Now, another question is around the off-target effect between a standard guide RNA and chRDNA. Love the name, by the way. So have you compared like, and I don't know how feasible it is, but the exact same sequence, obviously, not necessarily the same genes, but to really see, is it the modifications, or is it just the mechanism? I don't know if basically, is your guide RNA was exactly the same sequence between your standard guide and your chRDNA?
Mm-hmm. Yeah, unfortunately, I don't know the answer to that question. I believe that development is upstream of the process development group. Okay. I can say we try to create as close of an apples to apples comparison. The chRDNA development is very high throughput. I mean, we go through and just change nucleotides and create like a group, a cluster of them, and put them into an array, and just really pick. They will test, I think, in a 96-well format and see which one performs the best and pull that out from there.
Okay, so okay. And is the rule of design, like with the guide RNA, you have the PAM sequence and all of this, is it the same for the chRDNA, or they obey different rules?
I don't know the answer to that.
Sorry. It's probably the group upstream. Okay. So with that, let me see. Oh, another one for Evan. I don't know if you tried this. I've noticed, in your conditions you have IL-2, but have you tried to grow those T cells without IL-2? And what's the difference? What's the results if you've done it?
Yeah, well, at small scale, we've definitely done stuff like that in the G-Rex or flasks, and it's not very good. We do find that we can, obviously, we don't have to use IL-2. We have validated that we can use IL-15 and 7 instead of IL-2, which is often a big question as well. And then we can probably add cytokines less frequently in more static cultures. However, we haven't tried a bioreactor without IL-2 or without cytokine, but I would surmise it would be a terrible decision just based on the high fuel demand in those, especially when you get to higher densities and you need to really be supplying these, with food. But we haven't tried it in the larger scale, and the smaller scale doesn't do very well.
Okay. Let me see. Okay, so I don't see any more questions coming in. So maybe we're gonna close soon, a little bit earlier than scheduled. But so if you want more information about this talk and what's been discussed here, you can always contact your local Thermo Fisher representative. but also you can text CGT at 877-379-3018, and also go on our website at thermofisher.com/CGT. And we'll close this session. Thank you very much.